Hey guys, so today we're going to talk about a single synapse. When we're talking about a synapse, the first thing we need to know is what is a synapse? So, the synapse is the gap between the nerve cells, or a nerve cell and another cell, such as a muscle cell or a gland cell. If we look at this picture here, you can see there's two nerve cells right here, and there's a gap in between them. This gap is known as the synapse. And a synapse allows cells to communicate without actually touching or coming in contact with each other. So the synapse passes messages back and forth between two nerve cells, or a nerve cell with a muscle cell, or a nerve cell with a gland cell. So let's talk about the structure of a synapse. There's four really important parts of a synapse. We have the presynaptic terminal, the postsynaptic terminal, the synaptic cleft, which is the gap itself, and then we have neurotransmitters. And these neurotransmitters, we'll talk about them a little more in a second, but you can see they're inside these little vesicles inside the presynaptic terminal. When the vesicles start to move, we actually basically push out the neurotransmitters, and the neurotransmitters is what is the message. That's the message we're sending between the two cells here, between the presynaptic and the postsynaptic terminal. Okay, so we're going to break it down. We're going to talk about each part of the synapse. The presynaptic terminal contains the neurotransmitters inside the little vesicles. When triggered, the synaptic vesicles use exocytosis to push out the neurotransmitters into the synaptic cleft. Here, inside this cleft, or inside the gap here between the cells, the synapse, the neurotransmitters are going to activate the postsynaptic receptors, and those are these right here. So these receptors become activated when the neurotransmitters, if you can see in this picture, they connect. When, then we have the postsynaptic terminal. This is the receiving end of the communication. The presynaptic terminal has the message, and it sends it to the postsynaptic terminal, and they receive the message. This contains the receptor sites for the neurotransmitters, and those are right here, as you can see. The synaptic cleft is the space between the fibers. It's the connection between the neurons, and it's not actually a connection, it's a space, but it allows this flow of neurotransmitters and it allows these neurons to communicate without actually having a connection. The synaptic cleft helps the nerve impulses pass from one neuron to the other. Then we have the neurotransmitters. That's these little dots. We talked about them inside the vesicle. They get pushed out. They're the neurotransmitter receptors are on this postsynaptic terminal, but the neurotransmitters are chemical messengers. They have little messages, and they are sent from one cell to the other to pass the message along. These molecules are used by the nervous system to transmit the messages between neurons, or from the neurons to muscles, or vice versa. So let's take a quick second here to reflect on what we were just talking about, what we learned. So take a few moments to pause the video, read through these true and false questions, and when you're ready to go over the answers, unpause your video. Okay, welcome back. So let's go through these true and false questions really quick. Oops, sorry. Okay, sorry about that. I hit the wrong button there. So let's go over our answers. The first one says a synapse only transmits information between nerve cells, and that answer is false. It can transmit information between nerve cells with a muscle cell and nerve and gland cells as well. The second one says a synapse allows brain cells to communicate without touching. And that's absolutely true. The synapse is the gap between the cells so that the cells do not actually have to come in contact with each other. Number three, the presynaptic terminal contains neurotransmitters. And again, that is absolutely true. The presynaptic terminals have the vesicles filled with the neurotransmitters, and it sends the transmitters across the synapse, or the gap, to the postsynaptic terminal. And last but not least, number four, the neurotransmitters send messages between neurons and other cells. And that is true. The neurotransmitters are those chemical messengers, and they carry the messages between the cells for communication. So let's continue on talking about a synapse. So far, we've talked about what a synapse is and how it's structured, and now we're going to talk about the function of a synapse. So, a synapse has a couple functions. They are used to transmit impulses or signals. These signals can be chemical or electrical, and we'll talk about that more in just a bit. And the synapse is the action potential between neurons. So the synapse is actually the the way these neurons communicate. Without a synapse, the neurons have no communication. So the action potential is the act where the the chemical processes are actually taking place. So when we're talking about synaptic transmission, we have electrical transmission and chemical transmission. In an electrical transmission, the impulse occurs in the gap junction between the neurons. And in a chemical transmission, the chemical transmitters are passed through the junction between the nerve fiber and the muscle fiber, or between two nerve fibers. In an electrical transmission, sometimes the two nerves actually come pretty close to contact, so the gap junction is the space of the gap junction is decreased sometimes, and it actually sends electrical impulses through the gap junction. The chemical transmission sends the neurotransmitters, the chemicals between the junction. So let's talk about in terms of a synapse. How does alcohol affect this process? So ethanol exposure causes an influx in the number of chloride ions entering the cell, which lowers the membrane potential. And when the membrane potential is lowered, there's a decreased communication between the neurons. Intoxication is the behavior effects that result from the reduced signaling. So it takes a couple of tries before the signals to get across the synapse when the membrane potential is decreased. Remember, we just said a second ago that the synapse is the action potential between the neurons, the nerve cells. So if we decrease that action potential, then we're decreasing the communication. And so it takes a few tries for the signal to get across. And so that is why when people are intoxicated, they may have poor balance or slurred speech because the synapse isn't working properly. So the communication between the cells is being delayed because it takes 4 or 5 tries before it can actually get across the membrane. So when we're talking about the synaptic process, it all starts with a GABA receptor. And in a alcohol-free environment, the GABA neurotransmitter attaches to the receptor and it controls the cell's chloride ions. When it's exposed to ethanol, there is a influx in the number of chloride ions entering the cell, causing the potential to decrease. So that's why the action potential is decreased here, is because there's an increased amount of chloride ions that are entering. And so the lowered membrane potential gives us decreased communication. So there's a study, and the study is using the hippocampus cells of rats. And the study shows the long and short term effects that ethanol has on the synaptic process. And in this study, the control group is the the nerve cells that do not have any ethanol added to them. They're just observed in their normal environment. The experimental group is the nerve cells that have the ethanol added. And these scientists used the rat CA1 hippocampal subregion. That's the cells they were using for both in the study for both the control and the experimental group. So they hypothesized that alcohol will increase the activity of a synapse. And the results of their studies was that the ethanol increases the charge transfer of the postsynaptic currents. This increased frequency was maintained even after the ethanol was removed. So it's not just occurring while the ethanol is there, but it also leaves some lingering effects. So the study indicates um, that in the study, they used the calcium levels to indicate whether or not the ethanol had any effect. So, to directly assess the presynaptic terminal function, they measured the calcium transits in terminals in the neuron regions. So they're measuring the calcium inputs, and this image here shows the calcium entry is increased in presynaptic butons after exposure to ethanol. And so you can see here down here in this image, if the calcium level was increased at when the in the presence of in the presence of ethanol, which is the alcohol. So the study concludes that ethanol produces long-lasting potentiation of the 5-HT induced GABA release, and GABA was a receptor that is on the presynaptic terminals. Um, so it produces the long-lasting potentiation release by modulating calcium levels, and in turn, um, it it slows down the communication between your nerve cells, which leads to intoxication. Eventually, it will wear off. For some people, depending on the amount of alcohol you consumed, it may wear off within a couple hours. Depending on if the level is high enough, it could take um, upward to 10 hours for it to completely wear off and your synapse to be working properly. So let's take one last break here. And now you're going to do a quick write. So once again, I want you to pause the video. And once you pause the video, I want you to take 3 to 5 minutes to summarize the structure and function of a synapse and explain why synapses are important. So pause your video now, complete your quick write, and we are finished with our lesson today once you finish your quick write. So submit your quick write to me, and I will see you guys next time.